Hello friends, welcome back again. So today's topic is translation, that means protein synthesis. Chapter number 6, molecular basis of inheritance, translation. Okay, let's start. Translation natraga protein synthesis haibase kaino habase yengseko. Translation natraga protein synthesis haibadi process makhala mane mRNA dagi protein form tawbagi process makhala mane bako. It refers to the polymerization of amino acid to form a polypeptide. Amino acid se polymerization thoka polymerization sisu process makhala mane ko. Aduna amino acid se polymerize taraga polypeptide from the polypeptide haise kem nate protein dubune polypeptide haise ko. The triplet sequence of a base pair in mRNA defines the order and sequence of amino acid in a polypeptide chain. Triplet sequence of base pair haidi base pair laidana triplet sequence to. Triplet sequence genetic code to biniko man haise genetic code AUZ gaina ga laibado sigise triplet sequence ko yako. Sugumba triplet sequence se mayam se ko mRNA da mRNA aduna kai tawge haite polypeptide chain dugine order ama define tawebako aduga amino acid haidi polypeptide chain duda magi amino acid ki sequence su madu aduna piri nebako haibadi masigi haidi triplet sequencing sinako. It take place in ribosome. Translation se kadai da thoke haiba matam dane translation se ribosome da thueba. Aduna ribosome se cellular factory for protein synthesis haineba. Translation se kadai da occur tawge hai tardi ribosome da thueba. Aduna ni mRNA da thue hairi se ko messenger RNA da ko ribosome da thueba ko. Aduna cellular factory of protein synthesis hai tardi ribosome sineba ko si important ne cellular factory of protein synthesis high question lagtar di game nate ribosome net and means protein synthesis haibadi translation se kadai da thu e haiba matamda ribosome da thu e haigadaba ko. Next, important functions of ribosome during translation. Ribosome ki mathau se kaino ko translation tauri manungda ribosome ki mathau se kaino sine. Ribosome act as a site where protein synthesis take place from individual amino acid. Haibadi amino acid amadagi ne translation se haubase protein synthesis haubase ko ado ribosome haiba mafam sidagi hauba. Ribosome is a cellular factory haidana adudagi hauba ko habidi ribosome da yauriba amino acid to individual amino acid to adudagi haurineba protein synthesis se hana haubase ko. It is made up of two subunit anina semeba ko ribosome subunit anina. The smaller subunit come in contact with mRNA and form protein synthesizing complex. Ribosome ki subunit ani lay haibane ko ma haise ribosome ki subunit ani lay smaller subunit sina mRNA ga bind tawrani ko come in contact ha ko mRNA ga bind tawrani aduga protein synthesizing complex semani. Protein sem nangai complex sama semani bako protein do sem nangai complex semani whereas the smaller subunit act as an amino acid binding site whereas ko smaller subunit act as an amino acid binding site mana haise se large amino acid aduna smaller unit se ko amino acid binding site haibadi smaller subunit sina mRNA ga bind tawe contact come in contact taraga protein synthesizing complex ama seme haibane aduna sida smaller subunit act as an the smaller subunit act as an amino acid binding site haise si lale ko si larger subunit haigadamne ko si se moina lan bane si se larger subunit oigadamne smaller haise larger subunit act as an amino acid binding site amino acid nafam duna larger subunit ta oi haibane ba ko duka ribosome act as a catalyst for forming peptide bond. Catalyst haidi peptide bond nama form taunaba ko peptide bond that means protein synthesis taunaba polypeptide laidana adugi polypeptides peptide mayam dugi poly haise peptide mayam haibeni polypeptide haidi peptide bond peptide mayam hamne peptide singdugi marakta bond ama form tawrise si bond form tawrise ko si se si form tawhalise ribosome sina form tawhanmane ba for example oini yengbada 23 rRNA ribosomal RNA habene ko in bacteria act as ribozyme example oina hairabada bacteria gi oina ko 23 rRNA se sisna ribozyme oina act tawi ribozyme haise catalyst tu buneba ko ribozyme haise catalyst neba adudi yengse aduga translation gi process yengse kamaina translation se thokke ma gi process na kaino habisa ahan bada charging of tRNA charging of tRNA that means transfer RNA ko amino acylation of tRNA ko charging of tRNA natraga amino acylation of tRNA here amino acids are activated amino acids active ore amino acid plus ATP ko ATP ga amino acid ka pulaga amino acid se huzik activate tawre ko mathau to holani ba ko and linked to their cognate tRNA in the presence of amino acid tRNA synthesis aduga nga huziki active oiraba amino acid se tRNA ga link tawre ba ko adusu karamba catalyst yawi ko enzyme ma yawi ba enzyme ma yawi ba ko adusu in the presence of karamba enzyme amino acid tRNA synthesis size enzyme ne ko amino acids na tRNA da link tawba matam da ne ko punchan ba matam da karamba enzyme yawge haiba matam da amino acid tRNA synthesis yawi ba ko this process is commonly known as charging of tRNA or amino acylation of tRNA aduna simple oina sigi process sibu charging of tRNA ko charging of tRNA that means amino acid ka activate tawraba amino acid ka tRNA ga punchan basibu se punchin bagi process sibu amino acylation of tRNA natraga charging of tRNA ka weba ko adu sukaram be enzyme ma yawraga amino acid tRNA synthesis yawraga next process the initiation ka initiation the yin say translation is initiated by formation of an initiation complex consisting of 30s ribosomal subunit formylmethionine tRNA and mRNA translation se initiate ka karam kan tawge haida translation se initiate tare karam kanda formation of an initiation complex initiate bada sune posak ama change ba complex ama change ba ko complex ama change posak ma change ba adubu initiation complex ka weba ko aduda kai yawge hai tardi initiation translation se initiate tawna bo na bada change ba posak tu da kai yawge hai ba matam da 30s hai se ko subunit ko 30 subunit ribosomal se yawe ba aduga formylmethionine yawi tRNA gi ko formylmethionine yawi mRNA yawi si sinane translation process initiate tawrine hawrine ba ko 30s ribosomal subunit formylmethionine tRNA and mRNA it begins at the 5' end of mRNA in the presence of an initiation factor. Initiation factor haise ko initiation factor ga loina na ko karamba direction of the hauge matam da mRNA gi 5' end the hawi ba translation process ko adusu initiation factor yawraga the mRNA binds to the small subunit of ribosome AUZ is recognized by the initiator tRNA. mRNA se ribosome gi mRNA se ribosome gi small subunit ta bind tawre hanga sai gi ama habado subunit ani lei haidana ribosome gi larger subunit ama aduga smaller subunit sina mRNA da bind tawre haire ko aduga AUZ is recognized by the initiator tRNA ngasai AUZ se kai genetic code ne ko si methionine code tawrine mana methionine amino acid code tawrine ko yengo piriba haidi chart ama pibado genetic code ki chart ama pibado adu yawbane AUZ se karamba amino acid code tawge habdu ta methionine code tawbane si AUZ is recognized by the initiator tRNA transfer RNA na ne ko initiator natra transfer RNA se initiate tawna aduna AUG se recognize taweba initiation codon for methionine is AUG. AUG yare ko methionine ay nusi high kibado. AUG se initiation codon kaweba. Haidi translation se initiate tawbada masu yaudorine aduna ma se initiation codon kaweba. Translation process ta yauba initiation codon ki maming kay kauge habadi methionine kawe aduga magi code na kaino AUG ne ko. So methionine tRNA complex would have UAC at the anticodon site. Aduna initiation codon sina AUG oiramanina yengo to si AUG oiramanina magi anticodon di kayorani UAC orani mRNA ni ko mRNA ni A se adenine ga thymine ga base pair semidana ai hanjan mamang da swai from adenine ga thymine ga base pair se ya matam semgal adubu mRNA oiramnina mRNA oirabanina ko RNA oirabanina RNA gi oirabanina ne ko adenine gi mahutta ne ko adenine se thymine gi base pair semba gi mahutta uracil ga semakani ba aduna ne to magi anticodon se leire ko aduna ne magi anticodon se yengo AUG leire ko adu mai anticodon di adenine gidi kayorani U gidi A gidi uracil orani RNA oibagi ko U gina A oibirani aduga G gina C orani C na cytosine hairi ko adune magi anticodon se UAC lakani haise ko tRNA dadi next yense ko yengo zi next yense now the large subunit 50S bind to the small subunit to complete the initiation complex aduna huzikti ko huzikti large subunit ta ribosome gi subunit large subunit 50S small subunit ta bind tawsalani ba ko adu initiation complex to mapum phandoriba complete ohendoriba large subunit 70S has two binding sites to which the RNA carrying amino acid can bind haibadi yangoto subunit 70S ta binding site ani yauni ba ko aduna amino acid carry tawriba tRNA sina bind tawba yaba site ani yauni ba bind tawba yaba tRNA sina ne napsanba yaba ne site ani yauni ba ko 70S tadi one is called amino acyl tRNA binding site A site kawi aduga the other is called peptidyl site P site kawi amana 70S unit ta yauba binding site ani do amana A site kawi A site haibadi amino acyl tRNA binding site kawi amana P site kawi ko peptidyl site kawi ba ko next there is also a third site called the exit or E site where the tRNA are released ko haibadi adu site amasu lairiba ko third site amasu lairiba ani site ani haidi A site P site tudagi hendoktanane third site amasu lairiba adubu exit natraga E site kaweba ko aduda tRNA se release taraktoriba tRNA se next elongation haisna kaino yense ko the initiating tRNA carrying formal methionine binds to the P site haibadi initiating tRNA ko formal methionine carry tawriba tRNA sina ne P site ta bind tawrani ba P site ngasai P site to ko aduda bind tawni haidi peptidyl site to aduda bind tawrani ba another amino acyl tRNA complex with an appropriate amino acid enter the ribosome another atopa ne amino acyl tRNA complex to aduduna ne kadaida attach tawrani haiba matamda A site attach tawrani ba atopa amino acyl tRNA complex to aduduna ribosome da enter tawrani dana aduga a site amko attach tawbikhrani bako its anticodon bind to the second codon on the mRNA and a peptide bond is formed between first and second amino acid in presence of enzyme peptidyl transferase haibadi ko its anticodon du laidana ngasagi AUG ki anticodon do AUG ki anticodon do aduduna second codon binds to the second codon on the mRNA ki second codon du da bind tawrani ko and a peptide bond aduga peptide bond form tawrani first ka second ki amino acid ki marakta first ne second ne amino acid ki marakta peptide bond form tawrani adu peptide bond form tawba matamda da enzyme change ko karamba enzyme aduna peptidyl transferase changeba peptidyl transferase changeba ko next the ribosome now advances a distance of one codon and the tRNA that carried the formylmethionine is released at the E site haidi ribosome se hujikti ribosome se ko advances a distance of one codon and tRNA haibadi hujikti ribosome se haidi ribosome dagi lapthorakreba ko hujikti ribosome lapthorakreba codon duga tRNA ga codon se ko ribosome de lapthorakreba adudo kadai da thola ani haba matamda E site se exit site haba natra exit site da tRNA thola haba natra release tawre haba natra adu tRNA du da kai purake mana carry tawrake haba matamda formylmethionine se carry tawre a tRNA carrying the next amino acid now moves to the A site where the anticodon on the tRNA matches the codon on the mRNA tRNA se carrying the next amino acid tRNA se mathang haidi tRNA mathangi tRNA na ko haidi release tarakadaba tRNA se pura iba amino acid se hujik amuka A site ta move tawre and where the anticodon on the tRNA matches the codon on the mRNA haibadi tRNA gi anticodon ga codon ga ne match oire ko haibadi lairedan mRNA gi codon sina C neba AUG neba aduda mRNA gi codon si neba ko aduga tRNA sina codon neba mRNA gi ko AUG se mRNA gi codon neba ko si mRNA gi codon neba mRNA gi codon neba ko aduga magi anticodon se UAC sina tRNA da laibane be anticodon se tRNA na puyeba sigi anticodon se tRNA na puyeba adune mana haise adu match tawre ko match tawre habe samna habele nangna AUG se magi anticodon pio hairaga nang TACP bi yaroina si laleba ko si match tawdaba yaroiba match tawgadamne ba si aranbane ba ko aduna mana matching oi helle haise this is called translocation adubu translocation kawe habane ba ko the ribosome shift down by a distance of one codon hujikti ko ribosome se shift tawre ko distance of one codon amagi distance da ko shift tawre ribosome se mafam do mai mafam du dagi ko as the shift occurred the two amino acid on the tRNA in the P site are transferred to the new amino acid and the second tRNA is released from the E site hujikti ribosome se shift tawramnina ko mafam do change tawramnina amino acid ani do tRNA da P site ta lairiba tRNA na pura iba amino acid ani se ko aduna transfer to the new amino acid anobam da transfer tawreba ko aduga second tRNA sina E site ta release tawreba ahanbagi tRNA sina anauba amino acid da transfer tawrani aduga ani shuba tRNA sina ne E site ta release tawgadaba ko sise ꯍꯣꯏ ꯀꯩꯗꯧꯕꯗꯗꯤ ꯍꯥꯏꯗꯤ ꯈꯪꯄꯥ ꯄꯥꯕꯗꯗꯤ ꯒ꯭ꯌꯥꯟ ꯇꯥꯕ ꯋꯥꯅꯤꯕꯀꯣ ꯑꯗꯨ NCERTꯗ ꯑꯃꯨꯛ ꯍꯜꯂꯒ ꯄꯥꯎ ꯑꯗꯨꯒ ꯑꯩꯅ ꯇꯥꯛꯏꯁꯤꯁꯨ ꯇꯥꯎ ꯑꯗꯨꯒ ꯒ꯭ꯌꯥꯟ ꯑꯗꯨꯝ ꯇꯥꯔꯛꯀꯅꯤꯀꯣ ꯇ꯭ꯔꯥꯟꯁꯂꯦꯁꯟꯁꯦ ꯀꯩꯁꯨ ꯂꯩꯕ ꯅꯠꯇꯦ ꯌꯥꯝ ꯂꯥꯏꯕꯁꯤꯗꯤ ꯄ꯭ꯔꯣꯁꯦꯁ ꯃꯈꯜ ꯑꯃꯈꯛꯇꯅꯦꯕꯀꯣ the ribosome continues to move along the mRNA aduga hujik ribosome da shift tawkhrimna ribosome da hujik mRNA da move tawba hawrani ba ko chatpa hawra mRNA da lakpa hawrani ba ko aduga and new amino acid are added to the growing polypeptide chain hujik anauba amino acid ta aduduna polypeptide chain anauba hujik form tawba hawgala isa asida add tawrani ba a group of ribosomes associated with a single mRNA for translation is called polyribosome natraga polysome. Group of ribosomes associated with a single mRNA. Haibadi single mRNA magane ribosome gi group amagane translation haidi translation thokpada associate tawrise ko single mRNA ga group of ribosome ga adubu polyribosome natraga polysome ko polysome haibane ko polysome natraga polyribosome ko ba mathangda termination yengse termination se kaino haibase ko translation gi process termination elongation of polypeptide is terminated when a stop codon moves into the A side haibati elongate hujik polypeptide se elongate tawri ini dana ka chain sum semak pad elongate taw thola ineba adu do terminate se ko kanana tawge ha matam stop codon na tawi moves into the A side ta stop codon se move tawre dana aduga terminate tawrineba ko a stop codon does not specify an amino acid and does not have a corresponding tRNA stop codon dane amino acid amte yaude mangonda tRNA so yaude stop codon haise stop codon ne mangonda initiation codon so yaude amino acid amta su code tawde tRNA so yaude stop codon haise aduna termination haise loisan badubune bako haidi elongation chain sema iba aduga ngasagi mamangdegi chathariba process to terminate tawbu loisan badubune adubu kana loisan ge matamda stop codon loisan leba when amino acid tRNA reaches the termination codon like UAA, UAG, UGA, known as stop codon, the termination of translation occur. Haibati ngasagi amino acid tRNA sinane amino acid tRNA sina termination codon se yauraba ko haibati termination codon se da yauraba termination codon haidi stop codon do ne stop codon se ahum khatane lairi se stop codon genetic code ki chart to yengo stop codon ahum khatane piri se ko sise by heart of so important ne UAA UAG UGA sise bunye sina translation thokhan base termination tawhalleba loihalleba translation thokhan base the polypeptide and tRNA are released from the ribosome aduna polypeptide ka tRNA se ribosome da release tawhalleba ko ribosome dagi release tawhalle haibadi magi termination magi process to terminate tawra abanina adudi hujik ribosome dani dana thoise translation gi process na side translation process to is ribosome dani ba ko aduna ribosome gi process dane hujik exit site to dani ko release tawrak tori se aduna polypeptide ka tRNA polypeptide that means protein do buni ba ko polypeptide harangau nu protein do buni ba tRNA ga se ribosome dagi release tawra ani ba the ribosome dissociate into 50S and small 30S subunit at the end of protein synthesis. Ko protein synthesis ki process se aroi badane ko ribosome sinane 50S subunit amaga 30S subunit amaga se dissociate tawe ba ko and mRNA has an additional sequence mRNA se magi additional sequence leiba ko that are not translated mRNA ni na translate tawri se mRNA ne translate ngasai digi translation thoi se mRNA ne ba adu mafam na kadai de thoke ha matam na ribosome da ne ba thoi se ado magi sequence se tongan ba hen the sequence lei ba ko adu do translate taw de ba haidi adu de adugi mafam do protein synthesis taw ba ya de ba ko untranslated region ha ba ko andaga UTR haise untranslated region haise. UTRs are present at both 5' end before start codon and 3' end after stop codon. UTR se kadai the present oige haiba matam dane start codon haidi initiation codon gi mamang da yawe bako 5' end initiation 5' end ni dana ko se initiation codon gi mamang da yawe ba start codon gi mamang da ko aduga amana stop codon gi mamang da yawe sorry stop codon gi matung da yawe bako they are required for efficient translation process. That means moi da di translation thokte moi haidi UTR region se untranslated sigi region se di translation thokte haiba wane bako adubu translation translation akoina process tawrise adubu efficient oinabagi damakta di yamne haidi accurate oinabagi damakta di moi se yamna darkar oi haibani da ko sine ngasigi haidi ngasigi topic se ko translation haibidi protein synthesis hai se ko si loraga topic ahum khata ngai re ko DNA fingerprint ka human genome project ti aina ka shu thoina tak na ngai laite noi gi pa na ngai ne ba adubu ai haidi aina ngamba aina khanga da ba noina khanga da mong de explain do ai tauge ba ko aduna mayamna video se haidi gyan ta da ba khangdaba lairasu ai comment ta tum hairo o ai dum reply tarake ko ai dum clear tarake duna ai channel se like share subscribe to ba kaunu ko thank you